The ex vivo porcine keratitis model presented here provides researchers developing novel antimicrobials with a representative in vitro model to more accurately determine antimicrobial efficacy at the preclinical stages.
We have used Pseudomonas aeruginosa for this experiment, but the model also works well with other bacteria and organisms such as fungi and yeast.
Use sterile forceps to transfer the eyeball to a petri dish. Remove the conjunctiva and muscle tissue around the eyeball using a scalpel blade number 15 and forceps. Then, while holding the optic nerve with the forceps, gently lift the eyeball and transfer it to a half-liter jar filled with sterile PBS. Once all eyeballs are cleared of surrounding tissue, use sterile forceps to move them to another half-liter jar filled with 3% povidone iodine in PBS. After the eyeballs have been in the jar for 1 minute, transfer them to a third jar containing sterile PBS. Place an eyeball on a clean petri dish and use the forceps to hold it steady. With a number 10A scalpel blade, make a cut near the cornea. Use scissors to excise the cornea, leaving about 3 millimeters of sclera surrounding it. Ensure the sharp end of the scissors is in the suprachoroidal space and does not pierce the iris. Hold the corneoscleral button with forceps and use another pair of pointed end forceps.